Alright, biology, welcome back to video 10.2, part 2. Each pair of chromatids is attached at the center called the centromere. Centromeres are usually located near the middle of the chromatids, although some lie near the ends. A human body cell entering cell division contains 46 chromosomes, each of which consists of 2 chromatids. Alright, look, this is what this is saying. Basically, before cell division, you've got one copy of DNA, and DNA can be in the form of a chromosome. And this is kind of what this looks like. A chromosome, it looks like this shape with a center part called a centromere. However, when it enters cell division, the cell makes a copy of the chromosome, an exact copy, except my drawing isn't exact, and connects them in the middle with the centromeres. And in this case, these are called sister chromatids. This is a chromatid and this is a chromatid. They're exact copies of one chromosome. And when the human body cell goes into division, there are 46 of these. The cell cycle. At one time, biologists described the life of a cell as one cell division after another, separated by an in-between period of growth called interphase. That's a vocab word. Interphase is the in-between period of growth. We now appreciate that a great deal happens in the time between cell divisions and use a concept known as the cell cycle to represent reoccurring events in the life of a cell. The cell cycle is a series of events that cells go through as they divide. Another vocab word, cell cycle, a series of events that a cell goes through as they divide. Are you writing these down as vocab words, maybe on a flashcard or a folding piece of paper? During the cell cycle, a cell grows, prepares for division, and divides to form 2 daughter cells, each of which then begins the cycle again. The cell cycle is shown in figure 10-4. Okay, and here's 10-4 right here. So basically, this is the whole cycle. The cell growth happens first as a cell gets big, and then it comes to a point when it's ready to divide, so the DNA is copied, DNA replication, and when DNA replication is complete, it gets ready to prepare for mitosis, which is the separation, and then in mitosis we have 4 phases: prophase, metaphase, anaphase, telophase, and cytokinesis. Remember, we learned that mitosis is the separating what? The DNA, and cytokinesis is when the actual cytoplasm separates. Alright, let's keep reading. During the normal cell cycle, interphase can be quite long, whereas the process of cell division takes place quickly. Interphase is divided into 3 phases, G1, S, and G2. Okay, this is important, class. I want you to know the different phases of the cell cycle. I want you to know that the G1 phase is a period of activity in which the cells do most of their growing. During this phase, the cells increase in size and synthesize new proteins and organelles. G1 is followed by the S phase, in which chromosomes are replicated and synthesis of DNA molecules takes place. I want you to know that in the S phase, DNA is copied and new chromosomes are made. Key proteins associated with the chromosomes are also synthesized during the S phase. Usually, once a cell enters S phase and begins the replication of its chromosomes, it completes the rest of the cell cycle. And finally, when DNA replication is completed, the cell enters into G2 phase. The G2 is usually the shortest of the 3 phases of interphase. During the G2 phase, many of the organelles and molecules required for cell division are produced. When the events of this G2 phase are complete, the cell is ready to enter the M phase, which begins the process of cell division. Okay, so remember, we've got a couple phases. We've got the G1 phase, which is basically growth, getting the cell big and happy. We've got the S phase, which is copying its DNA. We've got the G2 phase, which is usually for helping. G2 is getting ready to divide. And finally, after G2 is the M phase, or metaphase. M phase, which is mitosis, which is actually doing the dividing. Alright, so let's go to the next video. Please go to page 246 and 247 for the next part of our learning.